Pesticide residues are frequently detected in our foods. This presents a lot of concern to consumers. Pesticides are effective because they have the ability to cause harm to pests such as weeds, insects, and plant diseases. And the fact that these same chemicals are showing up in our food supply is something that does cause a lot of consumer concern. In this task force report, I'll discuss some of the major findings that relate to how much exposure we have from pesticide residues in our foods and how these types of risks are assessed. Before launching into the highlights of this report, I certainly want to thank many people who were involved in the production of this particular report. Certainly want to give a shout out to my co-authors, Carol Burns and Steve Savage, as well as many of the reviewers who provided great feedback on this paper. And I want to thank the CAST group, both its board of directors and its support staff for helping this project become a reality. I want to start off by talking about how pesticides are regulated in foods. From there, I'll move into a discussion of how we assess the risks from pesticides in foods and what those risks might be in terms of consumer adverse effects. I'll talk a little bit about the concepts of epidemiology and biomonitoring, as well as pesticide benefits, and then I'll provide some concluding statements. The title of the task force report is Interpreting Pesticide Residues in Food. So let's start off by interpreting some residue data. So this particular slide demonstrates the results of the 2016 FDA Routine Marketplace Monitoring Program. In this program in 2016, FDA took 2,670 domestic samples and 4,276 imported samples and analyzed those for residues of approximately 200 pesticides. If you look on the left, you see the results from the domestic sampling. Of these, 52.9% of the samples showed no detectable residue, while 46.2% showed residues that were detected but were within the allowable levels, known as tolerances. And violations were detected in 0.9% of the case. Among the import samples, 50.7% of the samples had no detectable residues. 39.5% showed residues that were detected but within the allowable levels. And 9.8% of the samples showed violations. A good question is, what do these data mean? What can these data tell us about the potential risks from pesticide residues in food? And I've seen this type of data used by people that will say this data demonstrate that pesticides don't pose any risk at all to consumers, as well as other people who will say, if you look at this data, we see that the potential risk to consumers may be quite high from pesticide residues, and consumers should avoid consuming pesticide residues. So we'll get into a discussion as to what these data mean in just a bit, but before we do that, I want to talk a little bit about the regulation of pesticides. In 1996, the Food Quality Protection Act was passed unanimously by Congress and signed into law by President Clinton. And a major component of this particular act was that the EPA needed to demonstrate that consumer exposure to pesticides represented a reasonable certainty of no harm. If that wasn't the case, then pesticide uses would not be allowed to be made. And doing this, a pretty high bar was established. Not only did the traditional risk assessment practices need to be looked at, but the EPA also had to consider the potential impacts of the pesticides on infants and children, look at the aggregate exposure, so considering exposure to both food, water, and exposure in the residential environment, and also cumulative exposure when there was a case where a particular group of pesticides shared a common toxicological mechanism. In these cases, rather than look at the individual pesticides one by one, it was important that the entire family of these pesticides be evaluated for the potential risks. If a pesticide has a potential to leave a residue in a food item, then a tolerance needs to be established by the US EPA. Tolerances are set specific to both the pesticide and the commodity. So a particular pesticide might have different tolerances on different commodities. A particular commodity might have different tolerances set for different pesticides that might show up on it. Now, the EPA will only allow establishment of pesticide tolerances when it has been determined that the current and proposed uses of that particular pesticide represent a reasonable certainty of no harm, as I just described earlier. If that's the case, EPA then will set tolerances for that particular pesticide on the particular commodity. What is not well understood is the process by which these tolerances are established. Basically, provided that the EPA has determined that the pesticide poses a reasonable certainty of no harm, the tolerances are going to be set to be high enough to ensure that the user of the pesticide, when following the label requirements, so applying the pesticide on the proper commodities, applying the proper amount of the pesticide, harvesting the food after the minimum interval between the last application and harvest. So they will set the tolerance level to be at the high levels of what you would expect if the pesticide were being applied properly. So this is not a case where tolerances are set on the basis of safety. Safety has already been factored into in terms of the determination of the reasonable certainty of no harm, but tolerances are set to be indicators of good agricultural practices and not safety standards. If a producer is using a pesticide properly, residues of that pesticide should not be above the tolerance level. Let's talk a bit about what it means for a pesticide residue violation to be found. There are two major ways in which pesticides can result in violations. One of these is when the actual residue detected exceeds the established tolerance for that pesticide on that commodity. In general, this is a pretty rare occurrence. I showed the data that showed 0.9% of all the domestic samples had violations, according to the FDA in 2016. Of that 0.9% of the violations, 16% of those were cases where the residues exceeded the tolerance. In fact, 4 out of 25 violations had the residues exceeding the tolerance. And that's similar in the imports, where only 15% of the 9.8% of violations showed residues that actually exceeded the tolerance. The more common way for residues to be in violation is when they are determined on commodities at any level for which no tolerance had been established. So if a pesticide had drifted from an adjacent commodity to another one, or there had been uptake of contaminated soil, or there might be commingling of various foods at a packing plant, that could leave a particular residue of a pesticide on a commodity for which no tolerance had been established. If that is detected on that particular commodity, that's going to be a violation. So that's the primary way in which violations occur, not because residues exceed the tolerances. What does it mean toxicologically for a residue to be in violation? We talk about this quite a bit in the paper, but the general take-home point here is that residue violations rarely are of health concern. We could raise, actually, the tolerances to much higher levels and still have those levels be protective of public health. And we have several citations in which studies have pointed that out. Now, in some cases, that simply wouldn't be practical. The lower levels are already in place and force compliance with use regulations. So we do have a nice system to enforce compliance with that already. There's no reason to raise the allowable levels if we wanted to base them on safety. The important point here is that a violation does not mean an unsafe residue. Very rarely would pesticide residue violations be of health. Let's now return to the graph that I showed earlier and see if we can make something out of this. So a lot of the attention is often given to the disproportionate amount of violations in imports versus domestic. 9.8% of the imported food had violations, 0.9% of the domestic had violations. But what does that mean in terms of consumer health and safety? As I just pointed out, violative residues are very rarely of any health concern, so just citing this particular data does not really provide any useful information. Well, let's look at the blue lines. The blue lines represent cases where pesticide residues were detected, where the residues were within the allowable levels. 46.2% of the domestic samples, 39.5% of the imported samples had within the allowable levels or the tolerances. What does this mean in terms of consumer exposure to pesticides? To back up, we need to take some advice from this fellow in this slide. His name is Paracelsus. He's credited with being the first toxicologist. Back in Paracelsus wrote that all substances are poisons. There is none that is not a poison. The right dose differentiates a poison and a remedy. Or to paraphrase Paracelsus, it's the dose that makes the poison. It's the amount of a chemical, not its presence or absence, that determines the potential for harm. So I want to talk a little bit about how we determine what might be an acceptable level of exposure to a particular pesticide. So bear with me now as I talk about some toxicology jargon. One term that you'll often see is something called the reference dose, or RFD. This is defined as the level of daily exposure not expected to produce any health effects. So the goal of a good regulatory policy would be to make sure that consumers' exposure to pesticides is going to be below the reference dose level. We can set reference doses based upon the duration of the exposure. So for an individual pesticide, you could have a chronic reference dose, which would reflect the average level of exposure that you want to be below on a lifetime basis, or an acute reference dose, which would be the level of exposure you'd want to be below on a single exposure basis. Another important toxicological factor. We use this value then to calculate the reference dose. And we don't directly say the reference dose is going to be the no observed adverse effect level. We don't know, for example, how humans might relate to the most sensitive animal species that we've studied with respect to sensitivity to the chemical. It's possible that humans could be more sensitive to the chemical. So often we assume that humans are going to be 10 times more sensitive to the toxicological effects than the most sensitive animal species. We also don't know how the population in general might react to a particular chemical. And we might assume that there might be some members of the population that are more sensitive than the average members. So we often assume that some members of the population might be 10 times more sensitive than the average member of the population. So multiplying a 10-fold uncertainty factor from animal to human extrapolation with a 10-fold uncertainty factor from human to sensitive human, we come up with a 100-fold uncertainty factor. So for a lot of cases, we calculate the reference dose by identifying the NOAEL from animal toxicology studies and dividing it by a factor of 100. There may be other cases based upon perhaps data limitations or the quality of studies used to determine NOAEL. Or cases, for example, where infants and children might be more susceptible to pesticides than adults. An additional uncertainty factor, such as a 3-fold factor or a 10-fold factor, in addition to the normal 100-fold uncertainty factor that we use to determine what the reference dose is. So what level of exposure do we typically have from pesticide residues in the foods, and how does this level of exposure relate to reference doses? In the paper we cite this particular study that evaluated chronic dietary exposure to pesticide residues in the United States. This study looked at results from the FDA's 2004-2005 Total Diet Study. This is a market basket study in which FDA inspectors purchase foods at retail outlets in different geographical locations, presumably just as you and I would as consumers. Then these foods are prepared for consumption. At the time they're ready to be consumed, then they're analyzed for pesticide residues. When we understand what the typical, how much these individual foods consumers might eat, it's possible to come up with some crude predictions as to how much total exposure there is to those particular pesticides. In the 2004-2005 years, the FDA looked for residues of about 300 different pesticides. Of those, they did detect 77 pesticides were sampled in the total diet study. So in this particular paper, the exposure estimate for those exposure to the 77 reference dose values. So what did that study find? In 3 of the 77 pesticides, the exposures were greater than 1% of the reference dose. For 2 of these, they were between 1 and 2% of the reference dose. For another one, it was slightly higher, but the EPA has subsequently raised the reference dose for that particular chemical. So under today's evaluation, the exposure would be far less than the reference dose for that particular chemical. 14 pesticides showed exposures between 0.1 and 1% of the chronic reference dose. Another 19 pesticides had exposures between 0.01 and 0.1% of the reference dose. 41 pesticides that were detected had exposures less than 1/100th of a percent of the reference dose. So I'm throwing a lot of numbers out there. Let's see if we can put some of this in perspective with respect to the toxicology testing. An exposure of 1% of the reference dose, assuming a 100-fold uncertainty factor, as we went from the no observed adverse effect level to the reference dose, would represent an exposure 10,000 times lower than the no observed adverse effect level. An exposure of 0.1% of the reference dose would represent an exposure of 100,000 times lower than the no observed adverse effect level. An exposure of 0.01% of the reference dose represents an exposure 1 million times lower than the no adverse effect level. So for most of the pesticides detected in the total diet study in these years, was at least 1 million times lower than levels that don't cause the most sensitive toxicological effect in the most sensitive animal species. One conclusion from this is that our typical chronic exposure to pesticides is far lower than levels of health concern. You may often see media accounts of the results that look at epidemiology and biomonitoring. These studies look at patterns and causes of disease. We might know that one particular exposure to some agent might be linked to a particular adverse effect outcome. And we'll spend a bit of time on this, but these indicate correlations between specific conditions and health outcomes. When we talk about pesticides, there have been some studies that have indicated correlations between pesticide exposure and factors such as decreased sperm count, diminished IQ, and some increases in cancer. So these are studies that do get a lot of information. But let's try to understand a little bit better about what epidemiological studies can and or may not be able to tell us about the causality of some of these relationships. An important thing to consider here is that correlation does not mean cause and effect. For a lot of these studies, there may be a lot of confounding variables that might be more responsible for the particular health effects than something that's being studied. And we cite in the paper a French study that correlated French consumers that say they ate more organic foods with having a lower cancer risk. But in this study, there was no control for income levels, for exercise levels, or even for conventional fruit and vegetable intake levels. So it's possible that maybe a lot of other factors could have influenced the cancer risk, and it may not be due to avoiding pesticides by consuming an organic diet. We actually do find that even if you eat an organic diet, there's still some exposure to pesticides. It's generally lower than from conventional foods, but there is some exposure. And here's a bit of a facetious way to look at some of the studies that demonstrate correlation but not cause and effect. Here's a graph showing from 1965 to 1980 the pairs of brooding storks in Germany, and that compares very nicely with the millions of newborn babies in that same time period. Now, there's a great correlation here, but the cause and effect relationship, which would be the storks bringing the babies, and while that might be something that a lot of people consider as something that they're told as children, that probably doesn't really hold up. So we need to have some caution when we interpret some of these epidemiology studies. One good question to ask is, are the results consistent with other similar studies? If epidemiology shows that some particular activity is correlated with some adverse effect, How do other studies that have looked at similar things relate to that? Do you have consistency among those studies? If you do have consistency among the studies and they've been performed appropriately, that provides a lot stronger indication that perhaps there is something there in terms of possible cause and effect relationship. Another question, how accurate were the methods to establish exposure and or disease? I cite this French study we talked about moments ago, where consumers simply were filled out a survey and they were asked how much organic food they consumed. But it didn't look at all kinds of other aspects that might have also contributed to their potential incidence of cancer. And finally, is there a plausible biological mechanism by which the cause and effect could be postulated? If something is correlated with something else, can we break that down and to say, well, if this were actually being caused, what might be happening, say at a biological, biochemical basis? This could certainly stimulate certain research that might be able to elucidate possible cause and effect relationships better. So these are all important questions to ask when we interpret epidemiology findings. Finally, I want to wrap up by talking about the benefits from pesticides as well. We hear a lot about the potential risks. I've focused primarily on consumer risk, but there are also concerns about pesticides in the environment, pesticides in terms of occupational exposure. We do know that pesticides are used generally in concert with a lot of other types of pest control mechanisms, such as cultural, genetic, mechanical controls. But we do know that pesticides can help prevent crop loss due to pest damage, such as damage from plant diseases, insects, or weeds. What this means in fairly simple terms is that it allows us to distribute more food per area that we've grown. So reducing crop damage allows food. One thing we're all quite aware of from very strong epidemiological studies is that a diet rich in consumption of fruits, vegetables, and whole grains, whether they be conventional or organic, can reduce risks from certain types of cancers and heart disease. So if pesticides provide one way in which we can control pests, provide more food, make it more available, more abundant, more affordable for consumers, and consumers are purchasing those foods, then I think we have a chance for the benefits of pesticides to have a very positive impact on human health. My concern is that a lot of consumers are worried about pesticide residues in foods and might actually be reducing their consumption of fruits and vegetables because of their concerns. I cited the greater violations in imports. Well, we're normally encouraged to eat lots of fruits and vegetables throughout the year, but in our winter months, a lot of those fruits and vegetables are not produced domestically in the United States. So we often import a lot of those foods. And if consumers are worried about the imported foods being potentially hazardous to them and they consume fewer amounts of those, then that has the potential to some adverse effects. Or the worst thing they can do is not eat fruits and vegetables. There are also benefits to the agricultural pesticides as part of integrated pest management programs can stabilize against significant crop losses. You produce more food. This has a direct impact upon local economies that are depending upon agriculture. So you have the farmers, you have farm workers involved in that, you have people in food processing, food distribution that are all impacted by this. If you cannot produce a crop because there's too much crop damage, you waste a lot of food, and there's also, you lose a lot of jobs as a result of that. So it does provide some stable employment opportunities. I'd like to now conclude with some of our findings from the paper. One, very importantly, there is no direct scientific or medical evidence that indicates typical consumer pesticides result in health risks. A second conclusion is that epidemiological studies suggest correlations between pesticides and specific adverse effects, but often these studies are of questionable scientific validity. We need to really look hard at how these studies were conducted before we can properly assess their importance. And finally, pesticides provide benefits, and we need to consider those as well when we make regulatory decisions or we change agricultural practices. Thank you very much for your attention. I'd certainly be happy to provide additional feedback to folks. My email address is here on the paper. And if you are interested in downloading the complete paper, you can get it at www.cast-science.org/publications. Thank you very much for your attention.